Welcome, everybody. My name is Lammert Albers. I'm the business partner at GAP, overseeing our three lines of businesses. Firstly, our clinical trials business, the trial execution services business, where we help accelerate clinical trials for pharma, biotech, and medtech. Secondly, our GAP owned and operated site business, where we operate our own sites and help to accelerate the same clinical trials. And then thirdly, our blood and digital biomarker business, where we operate and drive insights on the blood and digital biomarker side of the business for all of our partners in an effort to accelerate the optimization of clinical trials and also help to improve delivery of tools for healthcare pathways as well. I feel very blessed today to be joined by two of our favorite partners at GAP. First, Rachel Radwan from Fujirebio. Fujirebio has been our partner both on Biohermes 1 as well as Biohermes 2. And of course, it's a well-known fact that Fujirebio was the first ever to have a clearance both on the CSF side of the house for an Alzheimer's test, and then also very recently for a plasma test as well on May 19th. Then secondly, we have Robin Wolz, who's the CSO for IXICO, who are our preferred imaging partner for both Biohermes 1 and 2. IXICO is the only central imaging partner that exclusively focuses on the neurospace. They have been involved in a number of very critical and important platform trials such as Amyloid, and obviously you'll have also supported us greatly on Biohermes 1 as well as Biohermes 2 now. So before we go into some opening remarks, I'll pass the mic to my colleagues here, Rachel and Robin, for some further introductions. So Rachel, why don't you go first?
Thanks, Lammert. Appreciate the opportunity to be here. I'm the Director of Clinical Research and Development at Fujirebio Diagnostics. And I'll pass that on to Robin. Thank you.
Great. Thanks, Lammert, for the introduction. Thanks for having us here. Robin Wolz, Chief Scientific Officer at IXICO. And as you said, we are a specialist imaging CRO focused on neuroscience, supporting primarily MRI and PET data collection in large multi-center clinical trials. And we work with pharma on their drug development, but we also increasingly work with diagnostic companies on approval of novel biomarkers, such as those from blood. So really looking forward to the conversation today.
Great. Well, thank you both. So today's talk will consist of a discussion really surrounding the key concept of advancing AD clinical trials through the joint effort between blood-based biomarkers and amyloid PET scans, so imaging. And so what we will do today is we will have a discussion amongst the panel, probably for about 20 to 30 minutes. We do have a chat function for the audience. I would encourage you to send questions to that chat function as you see fit. We will collect questions throughout, and we will start having a dialogue on that once we get a little bit deeper into the discussion. And then we will round up with some summaries as well. So the reason why we're here today is really as an extension of the partnership that was forged between our three companies here and the more global efforts that we pursued. GAP, during the time that we were facing a pandemic around the world, decided that we would start and deliver on a very ambitious project called Biohermes 1. And Biohermes 1 is a blood and digital biomarker study. And there were really two primary objectives of the program. First and foremost, we wanted to provide better tools and insights to optimize trial design of the future. And we were very lucky that we had so many terrific partners to the program that helped support that. And we were also very lucky that we were able to deliver such great data at such high rigor in a fairly short space of time. We ran this trial through the heart of COVID, and it took us 18 months to enroll 1,000 participants. And we were very adamant right from the start that we would run this study at such high rigor, meaning that we would make sure that all the data would be in CDISC format. We would have appropriate consent so that we would be able to use biospecimens for regulatory submissions. We then, of course, had the joy of working with these two partners here that we're talking to. I think the May 16th event with Fujirebio was probably the poster child of what we really wanted to get out of Biohermes 1. So 1, optimizing trial design of the future. And we're already seeing that play out now. We're starting to see a massive pivot in clinical trials where most clinical trials, particularly with disease-modifying therapies, are now adopting a biomarker-driven approach with regards to recruitment and enrollment. So we will touch on that subject as we go through today's discussion. And then secondly, of course, our second objective was a supportive and encouraging role to make sure that we are really pushing the field in order to get better tools out there for healthcare systems as well to diagnose patients. So the approval for Fujirebio was obviously a great success story, and we really hope that many will follow in the footsteps of Fujirebio. There was a very critical component in getting to that approval, and that's really the reason why Robin is here today to speak to that in some great detail. So I think with that, Rachel, it would be really great if we can pivot, and if you can speak to the audience and give us a run through, what does it take to really get through this approval process, start to finish? They say it takes a village. Talk to us about the village,
Sure. I'll try and keep it short. I'm not usually known for that. But it does take a village. The whole goal here was to work on this, having another tool in the kit to give something out there to help the patients, right? So currently, what was out there was for amyloid pathology is, we have PET and we have the CSF ratio. But again, looking at something that was a little less invasive, right? Trying to make sure that we get that out there and for use out there to, all the people, as well as for use and identifying it quickly for, now that we have more drugs available to use that with it. We here at Fujirebio worked really hard with FDA through their process with the breakthrough designation and the QSA process to work with them with what they needed, what they wanted. Again, to your point about your samples at Biohermes 1, a very important part and used within our submission, and very high rigor. The documentation, the data were the pieces that we knew we needed to use for the submission. Part of it as well that is that because of the CSF ratio and the PET tracers that had already been approved through FDA, we had to follow special controls to make sure that we performed or had performed an adjudication on the PET reads if we had samples who had used PET for the amyloid pathology. And so this is where IXICO comes in. And also everyone is aware of timelines, and, it's a business decision, how you meet timelines, how you can cut things without, cutting at your quality. And, the BioHermes 1 samples already had one visual PET read, right? And so with the adjudication process, through discussions with FDA and special controls, we had to do best 2 out of 3. So we then paired up with IXICO to go back, and BioHermes, of course, to allow us to get access and do this process again with them in order to get the adjudication performed for the PET samples that we had to have for that submission.
So Robin, we're passing the mic to you here. So, run us through your process in supporting this process, and, what did you face in terms of getting this done as a team?
Yeah, I mean, the most important thing, of course, is, if you start at the beginning, I guess it's having high quality data is of course critical, and you cannot do the analysis if there's an issue with the data. So bringing it back to what you described, Lammert, in terms of the setup of BioHermes and making sure all the processes are in place, everything is documented, everything is audit trail, so to speak, ready for inspection every time. And from our side, that involves from an imaging perspective, that involves making sure we have a harmonized image acquisition protocol, especially in multi-center studies like BioHermes. That's critical. So we make sure that the data that comes out of different scanners at different sites is of comparable quality, fulfills, of course, the requirements for the visual read, but also is comparable in terms of resolution and so on. We would then go through a formal site qualification process where that is rolled out to all the centers. All the staff are trained to collect data per that harmonized protocol, and then also make sure that we have an expert quality control in place where the data is inspected as it comes in to make sure that it's fit for quantitative analysis and visual read. So as Rachel and Lammert explained, obviously that was done as part of the BioHermes study. So when we entered the discussions with Fujirebio around looking at a second read and an adjudication, that was in place already, but is a fundamental and critical requirement to be able to run those kind of studies. As I guess Rachel alluded to, for the regulatory clearance, we require a gold standard of disease pathology in AD. That would currently be for amyloid, that would be CSF or PET. Here, of course, we talk about the PET. And I think Rachel touched a little bit on the requirements about robustness, reproducibility, and interpretability of the measurement, which is kind of like defined in the FDA in the relevant guidance documents. And so when we have high quality data in place, it's really around how can we analyze it in a robust, reproducible, and interpretable fashion. And to minimize any variability and also subjectivity, because we're talking about a visual interpretation by an expert here, we just need to make sure that we have robust processes and systems in place that make sure we can fulfill those technical requirements. And so what we do at IXICO, we have an end-to-end workflow that is integrated into our data management system, which is 21 CFR part compliant and in line with all the relevant regulations. And the data doesn't leave that system, so to speak. So the site would upload the data into that system. All the QC that we do is handled in the system, and then the visual read is also performed in the system, where the expert gets displayed an integrated viewer and a read form, which allows us to track and make audit compliant all the relevant steps in terms of the timestamps, the reader ID, the outcomes, any adjudication notes that come in place. And then, of course, readers are trained in a rigorous process to make sure we minimize any inter and intra-reader variability. And as we said, we finally, in this particular study, as it's of regulatory importance, the dual read and consensus approach is implemented, where 2 readers would read the image, and then a 3rd reader is involved for adjudication in discordant cases. There's also an interesting element in terms of how quantification and AI-based tools can support this process. What we have done in Biohermes is use what is called the VisQ process, which is a combination of visual and quantitative, where the expert reader gets presented output from a quantitative analysis and then uses that information to arrive at their final decision on positivity or negative. This actually was recently implemented or included in the in some of the tracer labels. So it has been acknowledged by the regulators that AI-based support can be a critical input into the read process, even though the final decision sits with the reader, and that's the process we have followed here. I think these are kind of like the different building blocks we need to make sure to run a study like that in a successful way. So making sure we collect good quality data and then have a robust and compliant process in place to analyze and document the output. What has been quite important in this particular study as well is the close working relationship and interaction with Fujirebio. So big thanks from our side to Rachel and the team, because we often, when decisions are made, and I'm sure Rachel can talk more to this, then timelines are critical, and then it needs to be done as quickly as possible. In a prospective study, collecting the data would be typically the rate limiting factor, but here the data was there, so it really came down to the teams working together closely to make sure we get the data onto the system, line up the readers, define the details in terms of the read paradigm, and then get the study running and provide the results back to the Fujirebio team, so it can be considered in statistical analysis and the discussion with the regulators. And so we had weekly calls with Fujirebio. There's a clear communication pathway that is put in place, a communication escalation plan to make sure that any roadblocks that come up are identified early and can be addressed and escalated quickly. And I think it has worked well here. And we managed to get through the data on time to support Fujirebio. So it's a great.
You did, and you did a great job. I think we definitely challenged your team with some of the thoughts, and because it was a different process than what you did originally for Biohermes, and in a prospective collection. But no, I agree with you. It was very collaborative, the weekly meetings, the conversations, and we had our timelines, obviously. And so very appreciative of that piece, but it was, and looking forward to definitely some more things to be working on.
Thanks. And we like to be challenged, and we like the difficult things. Otherwise,
worked out very well.
Exactly.
Mm-hmm.
So if I pivot for a minute, and if I just put on my sponsor hat, as many of you know, GAP is now running their 3rd sponsored own trial, Biohermes 2. We finished Biohermes 1, we finished Aphelia, and we're pretty close to full enrollment on Biohermes 2. There's some very interesting choke points. Besides the fact that GAP is now on their 3rd own trial, we have also supported hundreds of trials between our trial execution services business and our Goose, our GAP operated site business as well, predominantly or exclusively in the neuro field. And there are a few of these choke points that we have struggled with in the past, and we still struggle with. The problems surrounding PET are diverse and they're critical in nature. And they're not limited to selecting the right imaging vendor necessarily. It's upstream and downstream as well. So if we go upstream, there's a whole minefield of challenges with respect to getting access to ligands in the right locations and at the right cadence. And it can stop us in our track. We can be in situations where we activate sites on the premise that we're going to get access to ligands. And then once we are in site activated, we find out that there are some challenges with getting PET ligands. I think over the last 10 years or so, we've seen these problems ameliorate somewhat, particularly on the amyloid side. We're now living through those challenges on the tau side, particularly, whilst we're seeing, obviously a significant wave of anti-tau assets out there. So we're going to have to go through some of those same choke points. And so, like I had mentioned in the introduction, we're seeing a really quick pivot right now in our trials that we're supported GAP, and also trials that we hear about where folks are selecting a biomarker-driven approach and use blood-based biomarkers as a means to reduce some of the reliance on PET. And we're now starting to see some similar choke points, I would say, but of different nature. So, is it capacity? Is it turnaround of testing and the like? So, with that in mind, I'd love to now pivot the discussion a little bit and get somewhat operational with the two of you and tackle some of these questions. And by the way, I would encourage the audience to continue to send questions. We've got a number of questions already, but please feel free to keep sending them. We'll try and answer as many as we can in today's call. If we don't get to them all, we will provide some sort of summary with these answers. But one of the there's perhaps a controversial question, particularly for you, Robin. One of the questions we had when we started Biohermes 1 was an exam question around, well, is there a way to get to a world where we can take out PET? Can we take out PET scans altogether? We certainly have had quite a journey of our positioning with regards to that exam question, but I'd love to get your two views on that. Where are we in today's state? And what is the role for PET in clinical trials specifically? So, Robin, maybe you can start first, and then we can get Rachel's opinion on the matter as well.
Sure. Well, can we get rid of PET for clinical trials? My view certainly is no, at least not at this point. And happy to hear Rachel's thought on that. I guess I think for the foreseeable future, PET and CSF will probably remain from a regulatory perspective, the gold standard and in a way of biology or for Alzheimer's pathology. So there, and I think most workflows that are being proposed and implemented for blood-based biomarkers, and again, I'll let Rachel talk on that a little bit more, are using the two measurements in conjunction, where there will typically be a plasma test, and then you might screen out negative cases based on the plasma test, and then you have your indeterminate range that will go into a follow-up test with PET or CSF, and then the positives often would get confirmation from a PET or CSF as well. But that may change over time. There's other value that imaging remains to give, which is the spatial distribution that you will not have in plasma, and that is of course relevant for both amyloid and maybe even more tau, where we look at spreading longitudinally, as it can be critical for staging and also therapeutic monitoring. Obviously, as we think about amyloid removal, for example, we want to look at the PET scan to look at clearance. We see move towards surrogacy potentially, as amyloid PET has been considered a biomarker of surrogate endpoint of reasonable likelihood in all 3 approvals we've seen over the past year. So aducanumab, lecanemab, and also donanemab. So the role may increase there. And then last but not least, we will continue to use imaging most likely as a gold standard that enables further validation of those emerging modalities. So I'll hand to you, Rachel, before going into a lot more detail.
No, it's actually almost exactly what I was going to say. Right now, having the access for all 3 out there, but with our plasma ratio, once you hit that indeterminate zone, that is it, you need to confirm, and looking at that with PET and or CSF to see where that patient goes, what happens, what's going on with them, confirming, or if they do have amyloid pathology. The data showed that once you were positive, it was high enough, within that over 90% that it would be amyloid pathology, and then maybe moving them on into and identifying what drugs, right, and where they're moving on into that process. I don't think any of it's going to go away, right? It's about access. It's about, trust. Education is ongoing right now for plasma, blood-based biomarkers, and understanding all of these things can be used together to help treat the patients that are out there and get the drugs out to them sooner. So I think it's a combination of things. Down the road, maybe we'll have, as we see more and we have more data with the blood-based biomarkers, things may move to that because of the ease of it and the access to it. But I think at the same time, because of the way the stratification is with the indeterminate zone, there will still continue to be use of PET and CSF ratio.
Yeah, great. Well, and it's very aligned with how we see the world too now at GAP. But like I said, this is not how we started Biohermes. We really had this question like, how do we get rid of that big problem that we have? We now realize we just have to fix the problem in some cases. Rachel,
and I think, just if I can maybe comment on it, I think this is where we need to look at the potential of using those different measurements in combination, because you mentioned PET capacity as a bottleneck in trials, and there's obviously 2 types of issues there. One is availability of a certain ligand in a certain geography, and especially with novel tracers like tau PET, that can be an issue. And if it's not available in a certain geography, then you cannot fix that easily, right? But the other type of issue we're seeing is simply capacity, right? And we see, even if a tracer is available in a certain geography, it may not be available for all studies because there has to be prioritization and so on, and then there are production issues or bottlenecks. And I think this is where there's a huge potential if we combine the 2 measurements. So we make sure that we're focusing those PET doses or that we have on those subjects that need it, and where we cannot make a decision based on the blood-based biomarker in isolation.
Yeah, I agree, Robin. I think it's even if you talk about capacity, we even have remaining issues where in many locations around the world, just time on the scanner is an issue for neuroscans, right? So we may get ligand, we have a scanner, but the scanning facility is just so busy with oncology scans that there's barely any time to squeeze in a few neuroscans for research purposes. So it's like I said earlier, it's a very multifactorial challenge that GAP has now faced several times on their blood and digital biomarker programs. And we have to solve it. Every time we have a new ligand, we have to solve all of these different problems all together to get it done. Rachel, I'm going to put you on the spot a little. There's a very interesting question from the audience, which we've been toying with as well internally at GAP, partially out of intellectual curiosity, but also partly because of some inbound inquiries. GAP had been very disciplined. Our BioHermes team was particularly disciplined in the way we consented and collected biospecimens, and then we have a very disciplined way to control the pre-analytical conditions for said biospecimens, and we actually have an inventory of biospecimens still available for access for parties that have an interest in those plasma samples, whole blood samples, or PAX gene samples. And so the question we started to get on May 16 and beyond was, can you provide us access to the same samples that Fujirebio use? Because we're looking to go for a regulatory process. So the question we have from the audience is somewhat linked and related, and that is as follows. So the question states, do you think that other blood-based biomarker tests that follow you, Fujirebio, will be benchmarked against PET imaging, or is there going to be a world in the future where they're going to be benchmarked against Fujirebio's blood-based test? And I'd love it if you can give us some feedback or an opinion, what you saw back in the CSF days and what you anticipate in the future from a plasma perspective, Rachel.
So it's a great question. Something that we are looking at and facing currently. I think the first thing is that the next blood-based biomarker out there will still probably have to compare or to PET and CSF. There's no reference standard yet for these assays. And so you have to look at that as well, because, it's not just a method comparison, right? You are looking at the clinical validity in the population. I think I just said, not everybody's assay is made the same, right? So you have to take that into account and how it, and there's also guidances out there currently, looking at like the Alzheimer's guidances and different things that of reaching into, and what you're reaching for, of, your percentages, and depending on the intended use of your assay and what you're looking for it to do, right? Our plasma assay came up with the same intended use that it was for CSF and for PET. I mean, it was very powerful. The data shows it. But again, the next one, it will just depend. And that's also then when you're working with FDA and that conversation on that process, which, through the QSub process, you can ask those questions and figure out the best path with them.
Yeah. So from the same audience member, we have another interesting question, and very curious to hear your thoughts on this. How critical do you think it's to be first to get an FDA clearance? How critical is that to the commercial success? Any thoughts on that from
a commercial point of view, of course, first too is also very critical to the commercial success. And it's heartwarming. Like for me, the patient is first to me in the work that we do. We felt it with the CSF ratio, and now with the blood-based, with the plasma ratio. I think we're seeing it a bit more with the plasma ratio, because it is easier access for a venipuncture than it is for a lumbar puncture, right? But yeah, I mean, people have not always been first to market and been very successful. But I will say that for us, that first to market has been. And so hopefully there'll be a few more down the road. So
good. So as we're finding out and always expected, this is a very heterogeneous disease, and that's another reason why we really decided to cram as many digital tools as well as blood-based or wet biomarkers into our BioHermes programming, both 1 and 2. So if you, and this is to both of you, if you think about the evolution of the space going forward, how do you think about that, both from a digital and imaging, as well as blood marker perspective? So Robin, maybe you can have a stab at this one first, and then we pivot back to blood tests with Rachel. So what's your position on this, Robin? How do you guys think about that in IXICO, the heterogeneity of the disease?
Yeah, I mean, firstly, it's great that we have an increasing number of measurements at a more advanced stage of validation. We're looking at co-pathologies. We're not focused on amyloid and tau anymore. I mean, I was reflecting on this the other day in the context of other diseases like Parkinson's disease or Huntington's disease, where we're now getting to a stage of a biological definition, which we have in Alzheimer's for many years, and well-defined diagnostic criteria, and that's reflected in clinical trials for many years. And it's great to see how we're almost moving on from that and looking at the next wave of measurements that help us better characterize the population. And from our side, one thing that we're very interested in is vascular pathology, for example, and vascular, cerebrovascular disease as a co-pathology in Alzheimer's populations, where we're seeing in probably 50 to 80% of AD cases a level of vascular pathology, and that's MRI is the gold standard assessment there at the moment, and that's an area we're quite focused on, because it will help to having this multifaceted view on pathology will help to put things into context as we validate different drugs with different mechanism. If we have an anti-amyloid therapy, but assessing it in a subject or in a population that predominantly is affected by vascular pathology as a cause of clinical dementia, then the effect must be different than in one that is predominantly affected through amyloid pathology. And that's just one example. I'm sure Rachel can talk a little bit to the kind of different measures you're now looking at from a plasma perspective that will also help complement the picture here.
Thanks, Robin. Yeah, so the technology as it increases also helps us, because part of our submissions and the data that we are required to submit to FDA to support the intended use, right? So in the current one, it was, signs and symptoms, patients with cognitive decline. You obviously have to provide that information, what shows that they had cognitive decline. So again, the cognitive functional tests, and that different, we had MMSE, we had FAQ, and so on, and different ones from, our cohorts. But again, from these other technologies, as that speeds it up, it also adds in some rigor in there, because then it's a bit more standardized across, right? Than individuals using the same from that technology, and the same process going through. It takes out some of that subjective pieces similar to the adjudication that we talked about earlier, by adding that in there, and then, increasing that rigor and that data that's used for those pieces. Did I not answer your question, Robin? I think I was more on the other one from Lammert. Sorry.
Yeah, well, let's talk about rigor for a minute. And you guys are both on the cutting edge of science, and you're going for this innovation curve. Yet many of the audience today are recipients or beneficiaries of your technology, right? In the trials that they execute and deliver on. And so when you go for this innovation curve, you have always got a very interesting cost picture as well, where you start with high cost, and then you go into a commoditization phase of the technology, and for some technologies, that path to commoditization from a cost perspective is much faster than for other technologies. But it's obviously for the audience, particularly for pharma, biotech, and medtech, it's a very important, impactful thing to consider. Do I go for the best test that gives me the highest quality? Do I go with one that's good enough? Or is cost becoming a gating factor for me in terms of being able to operate my trials? So I'd love to get both your perspectives on that. How do we balance rigor of a test or a biomarker with efficiency and cost? How do you make those no-go decisions? And how do you guys think that the market is going to evolve in terms of the cost-benefit ratio with these types of biomarkers? So Robin, I don't know if you want to have a stab first.
Happy to go first. I think for us, one key question is, of course, to what extent can we reduce the human element and the human factor in the process. We talked a lot about visual reads, and we had even 2 or 3 in this instance. We did talk about the fact that they're AI supported insofar that the reader sees a quantitative output. And I think further pushing the boundaries in terms of what decisions can be made automatically by an algorithm as opposed to require a visual assessment, that's probably the way where we see opportunity to streamline things. And I don't think we're going to be in a world soon where it's fully AI based and completely automated. However, making decisions on clear-cut cases is something we're seeing is happening in clinical trials already. We're saying, okay, if you have a certain centiloid value, so quantitative analysis result of your PET is above a clear threshold or below a clear threshold, you're in or out. So very similar to the way the blood-based tests operate now, where you have this indeterminate range where you then have a follow-up, and in this case, it would be a visual assessment. So I think that's the opportunity we see, and the same applies to MRI as well. I talked about vascular markers earlier, where we see the same thing, where we see increasing the AI, for example, helping to support in detection of microbleeds or assessment of white matter hyperintensities, and so on.
So Rachel, there's to sort of pull on that thread a little. Do you think that Fujirebio and their peers, that are you guys still competing on best AUC and best NPV and PPV, or are we beyond that now? The way we look at the world right now, we had pretty much every 217 available in BH1. And everybody seems to be pretty close to each other. So our view of the world is perhaps a little different, and it's a bit more operationally oriented in terms of, well, what can you get and what are the costs associated to that? But how do you guys view? How do you think that Fujirebio and your peers? How are you guys going to compete for market share? Is there still an element of, hey, I have a better test, or are we in that space where we just want to be non-inferior to Fujirebio or non-inferior to ABC?
Oh, Lammert. So, part of Fujirebio's business model is, putting it out there, putting the product out there, proof of concept. It is known that we don't have the footprint out, worldwide. And so then we, once do this, we work with our partners to help them develop the assay and then work through the regulatory process to get that product out onto the market. You know, the goal here is also with our partners who have the larger instrument footprint, that we can get that out there and get more patient access. You know, in the end, that's the ultimate goal. Wait, go back to your question. Wait, say your question again about the commercial, right? So you know, for me, it's supporting that business on our end, getting it out there, and then, you know, pushing it through. But the cost, you're killing me here. Repeat the question. Sorry.
It's basically how do you think, you know, now that you guys are cleared, there are other fast followers. You know, if you go back in time, even 2 years, you know, it's clear that people were competing on the stage in terms of NPV, PPV, AUC. You know, I've got a better test than yours, and it feels like the playing field has leveled, and we're at a stage where everybody is at an acceptable level in terms of quality of the actual test, scientifically speaking. So is there still space to grow? Is there still, you know, can we still get better at this particular test, for example? Or do you think, you know, you guys as a peer group will compete differently, i.e. scale, cost, turnaround time of testing, with relationships that you might have with central laboratories and the like? Just interesting to see how you guys view the world. Is there going to be a next gen, for example, that's even better than the first gen?
Well, you know, there's always room for improvement. Don't get me wrong, right? Always looking at that, and let's be honest, what are we, 5 months in here? So I can't, you know, talk about the immediate. You know, we're already looking at the next gen and so on. You know, we're just working through right now what's happening. You know, with 2 years ago, everyone was talking about 217 and, oh my gosh, it's this thing, and now, you know, we've done this, and now it's another marker, right? It's this quick turnover from one thing to another, because there is so much changing and happening in this space all the time, and understanding, you know, what p-tau 217 is identifying, or what's it showing, what can it do. So it's a little hard to kind of put that to answer that question at this moment. But again, number one, things can always get better, like working down, you know, in that process. But at this point, no, there's no next gen at this point. We're working, you know, with our product as is. So I can't answer that one.
So, so what do you guys think we need just to scale now? So we've got a test, Robin, on the imaging side. You know, we've got approved ligands, we've got great partners. You know, what do we really need to really scale this now that we have the tools available? You know, you know, I've mentioned a few times we're starting to see some challenges on the blood side, where we're waiting for result, which, you know, holds up sites, screening participants, for example. So how do we really sort of, you know, deal with all the scaling issues in the short term, so that, you know, we can get access to these tests? Or, you know, how do we continue to scale, Robin, on the imaging side, so that either blood markers or imaging is not on the critical path in trials? Really interested, you know, like, you know, how you see the world, Rachel, from a blood marker perspective. What are the big ticket items or small ticket items that we need to deal with to really sort of you know, crush choke points in terms of capacity? And then, Robin, for you, very much the same on the imaging side. So
Who you want to go first?
I'll go. Sorry, real quick. So I think part of that has to do with the study design, right? And the intended use, and what you're trying to get at, and who you're trying to get at. Because sometimes it's very, very specific, you know, inclusion, exclusion, what you're looking at and what you're doing. Study designs for diagnostics are obviously a bit more pared down than they are in the pharmaceutical biotech world. So I think it just maybe becomes more of a collaborative approach between, pharma companies and the diagnostic groups, and if they want to use it, what's best, what works, and re-examining that study design, or at least the intended use, or I'm sorry, the inclusion-exclusion criteria. So maybe that doesn't provide give you that pinch point that you have with enrolling those patients.
Yeah, and I think, we can talk about what each measure can do to become more widely available or build capacity, and there are obvious things, and some of those we touched on. I think the most interesting piece is, though, how do we integrate the different measurements most effectively to reduce bottlenecks, to increase efficiency, and to focus that. We talked about the fact that focusing the PET capacity on those subjects that really need it is to me seems like an easy way of, even with current distribution networks and all the caveats we discussed, free up capacity, right? And that comes back to making sure that, and I think it's what Rachel said as well, making sure that we set up a trial design and operational processes that allow those different tests to go hand in hand, to make sure we can quickly interpret the blood test to then quickly make a decision on who needs follow-up, and efficiently schedule that, make sure we only schedule it for those subjects that really need it, and then get the final conclusion quickly. In parallel, of course, we need to increase, distribution networks, especially for less established tracers. We talked about tau in principle, but even within tau, there's a difference between what people call first and second generation tracers, so those more sensitive tracers that most companies now want to use. They're even more difficult to access, and so working on that in parallel, I think, is critical. And I'm sure there's probably some steps that the blood-based biomarkers need to go through in terms of making sure we have all the standardization and all the analysis that can be done at speed. I'm not sure, Rachel, if you want to comment on that a little bit more.
No, you're absolutely right. There are different processes that we have to go through on that to, through the standardization, analytical, and validation performance. The one thing I was just thinking about is, I'm sorry, I lost it. In that, for the processes with the blood-based biomarkers, and currently they're out there at multiple labs, and that so that is out there. That show point can, at multiple the centers, at the labs. So it's clear. There's a piece about this that our regulatory submission and the data that we submit for a regulatory submission for the the clearance that we worked on for our ratio is a bit different than what's being used or possibly used for a pharma trial. So you also have to look at that and where it's being used, and that process. So.
Well, I feel like we can keep talking for hours, but sadly we're approaching the end of this hour, and at the risk of having people leave for the next meetings, I think we should seek to wrap this discussion up a little. I certainly hope we have another opportunity to get together here with the three of us and to continue this discussion. But I really hope that the audience has come to realize that there's been a terrific collaboration, and from what we at GAP at least perceived as being somewhat competing modalities, we very quickly learned that there is almost a symbiotic relationship between these, what appear to be symbiotic relationships. And over time, we've seen many partnerships being forged between digital, non-digital, wet and digital, and wet and imaging, and we just can't wait to see, you know, what the future holds and how more of these partners come together and create tools and insights for everybody, whether that's, you know, on the clinical trial side or on the healthcare side. And so, you know, that is my hope, and that's GAP's hope, that what we'd like to see in the future, you know, more of these partnerships. And maybe we can just conclude the discussion with, you know, what you guys hope for respectively as well to see in the future. Robin, so if you want to go first, and then we go to Rachel.
Yeah, I'm looking forward to working with, you know, Fujirebio and others in the field on getting this comprehensive picture of biomarkers that we need to understand each patient and how they fit into trials and how they're best diagnosed. So to me, it's really around bringing all these things together and using the most meaningful and most appropriate measure in the right way, and that's what I'm looking forward to.
Thanks, Robin.
Thanks, Lammert. Yeah, similar. Also challenging each other, right? The knowledge that we bring, the knowledge that Robin brings, and discussing it and understanding it, and how best we can present it back to uses for that, be it in clinical trials, or to the physicians who can use that within their toolkit to help these patients, you know, because as you said, there are multiple things, co-pathologies and different things, and new, you know, biomarkers every day pop up, and someone says, hey, did you hear about this? And how does that react, or how does that work? So, I mean, it's always changing, but this relationship, at least we can, will be collaborative, and we can work together to understand these things in the future for other projects.
Well, thank you both, Rachel, Robin. It was an absolute pleasure and delight to spend the hour with you, as always. And I would also like to thank the audience. If we didn't get to your questions, we will endeavor to provide a response and send those to you. And if you have any questions that come up as an audience after the fact, then don't be a stranger. Reach out to either of us or all of us. And once again, thank you, everybody. Thanks for dialing in, and talk to you soon. Thank you. Bye.
Thank you.
Thank you.